So we're going to do a little bit more on this concept of linked genes. So remember, Mendel was under the was under the impression that all genes assorted independently because he didn't understand chromosomes and the chromosome theory. Now that we understand chromosomes, we know that some genes that occur on the same chromosome are not independently assorted. And so that means that they will not follow the typical Punnett square ratios. Occur on the same chromosome. So essentially what that means is they cannot separate from each other in meiosis. They travel with the same chromosome, at least most of the time. We'll talk about that in a minute. You'll remember that there is that idea of crossing over, where certain chromatids sort of get very close to each other when they're in that tetrad form, lined up, ready to go in meiosis, and bits and pieces can transfer from one to the other. So what that means is a gene that's on the same chromosome might transfer to a different chromosome, and so there is some change that can occur. Linked genes don't always stay linked, but when they are linked and occur on the same chromosome, what that means is the Punnett square ratios, the Punnett squares that we do and the ratios we do will not apply to those. So you'll remember how if we took a particular flower. So for example, let's just take this example. This is from your textbook. There's a purple flower. And we're crossing it with a red flower. I'm just going to draw the actual flowers rather than all the stems and leaves and everything. And then I'll color them in red. There's a red flower. Well, actually, it doesn't look much like a flower, does it? Until I... And a purple flower. So I'll just make them look like flowers real quick. There we go. One flower. Another flower. Okay. And if we cross these flowers, and we use the dominant recessive idea for colors, there are two sort of traits that we can track here. Purple flowers and long pollen. So the pollen grains come out to be very long as opposed to these little sort of rounded pollen grains. Those are two traits that these flowers have. And so if we take a homozygous dominant flower in both cases and cross it with the recessive condition, right? What we would expect to get in the first cross are genotypes of heterozygous. That's the only real possible combination because you're only producing one allele in each case. You know what I'll do is make these L's little scripted L's so they stand out a little better. All right? Then what happens is we end up with all purple flowers. So the purple is the dominant color, right? So in the F1 generation, I'm just going to write all purple. But they have this genotype. So they're now heterozygous because of the cross. Then what we do is we cross the F2 generation. So we take those flowers and we cross them again. And what we find is that when we cross the next generation, we get purple flowers, some purple and some red. So in other words, here's purple and long. Purple flowers with long pollen. And we would get purple flowers with round pollen. Then we would get red flowers with long pollen. and we would get red flowers. Whoops, I wrote pollen instead of, hang on a second. Oh, why does that jump every time I do that? I don't know why. Back. Long pollen is what I meant to write there, and red with round pollen. But when we do the cross in a Punnett square, we get very specific ratios of what should happen, right? We figured all that out, and we should get, 3 out of 16, and we know all the different ratios that are expected. However, what we find is we get 15. Only 6 out of the 15, let me write it the other way. 6 out of 15 have this phenotype. And let's say we find, actually, sorry, I'm reading it wrong. Let me get this, here we go. Well, we find a 15.6, it wasn't a ratio, just a number. Then we get 1 like this, we get 1.4 like this, and 4.5, right? On average, these are the average outcomes. And they don't fit. They don't fit the Punnett square that would be resulted from these crosses. So they don't match. So we can't use a Punnett square to figure these out. What makes it difficult is that there is a way to figure them out, but it requires some pretty advanced research, and every different linked gene has different ratios in which it actually goes together. So we're not going to learn how to figure out these. That's something we'll leave to your studies later on in genetics. But all we need to know is that if you end up getting ratios that don't match the Punnett squares, so when you cross these animals or plants, if the offspring don't match what you would predict by drawing the Punnett square, you can tell that the genes might be linked, so they're on the same chromosome. And that's really the only issue. Now I want to talk about a specific kind of linkage, which is called sex-linked chromosomes. Sex-linked chromosomes are when genes occur on the X and Y chromosomes. So when we do this, what we have are, okay, so your X chromosome, kind of, I'll draw it in its doubled form that we see ready for meiosis, because that's how it has the X shape. And the Y chromosome, sort of doubled up like this, right? Joined in the middle there. There are lots of genes on here. There could be a gene up here, right? There could be another gene down here. There are literally thousands of genes on these chromosomes. So not only do they contain the genes that determine boy or girl, they also contain many other different genes. What you'll notice is that the homologous Y chromosome seems to be missing the bottom half. What that means is the top part is homologous to the top here, so it would have those same sort of alleles, maybe different alleles, but the same sort of genetic complement. But look what happens on the bottom. All of the genes that occur here would only occur once if you were a boy. Being a boy has a cost. And the cost is that all the genes on the lower part of this chromosome do not have a homologous partner. So that means there's only one allele that's determining whether or not you have a condition. So you don't get the advantage. If you are a female, right? Here's a female with her 2X chromosomes. Well, if you have a recessive allele here, and maybe that recessive allele is for some kind of serious condition. There are lots of sex-linked traits in humans, things like muscular dystrophy. Well, if you have an allele for muscular dystrophy here, and it's a recessive allele, if you're a female, you might have the same allele over here, or the same gene, but you might get the dominant allele, which would mask it, and therefore you would be okay. You wouldn't have muscular dystrophy. But if you're a male, guess what? You don't have that option. And so one single allele for muscular dystrophy, whoops, I erased it right there, is all it would take for you to have that condition. So because of the Y chromosome and being so much smaller than the X and missing some of the alleles, the homologous alleles, for many things, men have a kind of a bit of a disadvantage genetically. I'm going to give you a couple of examples. Now, it may not be a bad thing. Sometimes it's just something simple. So we've noticed, for instance, in fruit flies, we have red eyes and white eyes, along with male and female. So if we track, let's look at fruit flies here. Fruit flies are nice to study because they also have an X and a Y chromosome, very similar to the way humans do. So if you have a fruit fly, then here are the possible alleles that you can get. You can have an X chromosome that carries a red eye gene, right? You can have an X chromosome that carries the recessive version, which is a white eye gene. So this is the red eye, and this is the white eye. Now, the Y chromosome does not carry either one. So the I gene would be located on the bottom part of the X. The Y gene does not have an allele for this. Or I say the Y gene. The Y chromosome does not have an allele. So look at the possible combinations you could get. If you were to cross and get this combination, two X's, the two X's means you're female, and the two R's mean that you'd have a red eye. The other possibility is for a female to have one of each of these, which would mean you're still a female, two X's, and you would have still red eyes because of the dominant allele. That one allele right here is still dominant, so your phenotype is still red eyes. If we do another combination, we could do the two recessive possibilities, like this. This would mean you're still a female, but you would have white eyes because you don't carry the dominant allele. So there's three different possible ways that you can sort of end up if you're a female. And if we look at the percentages and chances, which we can do in a minute, we can see that there would be different possibilities. Now, if you're a male, let me just put the male over here in this box. You have to have X and Y. So there's really only two options for the male. I'll do the males in red. X with a big R and a Y, or X with a small r and a Y. And of course, the phenotypes that go with these, you would be male because you have X and Y, and here you would have red eyes, and here, male and white, because you only have the one allele. So whatever one you get, there's no dominant one to mask it. And of course, we can do crosses. We'll do an example of a cross, but those are all the possible outcomes, right? The possible outcomes. So let's do a couple of crosses. Let's take, we'll call these example crosses. Let's take a female who is homozygous for the red eye, and let's cross that. The X is kind of a little bit, I was going to put an X for cross, but maybe I'll put a plus this time. So we'll mate that with a female who is a male. This is a white-eyed male, right? So we have a red-eyed female, homozygous, and white-eyed, sorry, we have a red-eyed female homozygous, and we have a white-eyed male. Okay. So again, if we go through the process and look at the possible gametes that can be produced, the female can only produce one gamete. But the male, of course, can produce two different gametes, the X with the recessive R or the Y, when those separate in meiosis. So what we have to do is we have to look at the possible outcomes that we would get. So with only this possible outcome, we would see that the result would be red-eyed females, but they would now be heterozygous for the red eye, or red-eyed males. There's no way to get a white-eyed anybody in this particular cross. The only way to get a white eye would be if you had a white-eyed male and it was being mated with, well, actually, you could get a white eye, as long as the female is carrying the gene, I think it would work. So let's do this example. Let's look at a female who carries the gene. In other words, she's still red-eyed, she has that recessive allele, right? And then we cross that with a normal male who's red-eyed. Okay? At first glance, you might think, well, because the male only has the red dominant eye gene, everything should work out, that should mask and we shouldn't see any white. But because there's nothing on the Y chromosome, look what happens. We get an instance where we should find some cases here. Here we have the two possible gametes that could be produced by this particular individual, and two possible gametes that can be produced by the male. So if we draw a Punnett square, it's a little bit bigger this time. So we have the XR, there's our female, and here's our male on top. Oops, what happened there? We will get this combination, which is the homozygous red-eyed female, completely normal and not carrying anything. We might get female carriers of the white-eye gene, and then we will get some normal males, but there is also a possibility right here of getting a white-eyed male. So even though none of the parents were white-eyed, and even though the male carried the dominant gene, it's this one of the females, which isn't matched by the Y chromosome right here, which gives us the condition of the male having the white eyes. And you could look at all the different possible crosses. And you could figure things out. For instance, let's say that muscular dystrophy was something that runs in the family. So two parents want to know, for instance, what are the chances of their child having muscular dystrophy, right? And so if we were to do that, we could do a cross, let's do a human cross here. A human example, muscular dystrophy, which is a disease where your muscles basically don't function. They slowly degrade. Dystrophy means they stop working. They just don't work properly, and they weaken over time. So it's not a very good disease to have, if any disease is good. So let's take a human, and let's say that we have a female, right? And we have a male. We'll put them here. Now let's say that the male has no history of muscular dystrophy in their family at all. None. Right? They would probably have the healthy, I'll use a capital M, for the regular or normal gene. I don't think it's a dominant gene for muscular dystrophy. Let's say the female we're not sure. She seems healthy, but we don't know if she's a carrier or not. So we could go back in her family tree and we could find out that there were instances of people in her family who had muscular dystrophy. So what we might have to assume, if she was normal, we know that no problem, right? But if we assume that maybe she's carrying muscular dystrophy because it occurred in her family in generations past, and we did the cross, we could figure out sort of what the chances might be. So it's the same cross as we did before, just with different letters. The female can produce the normal allele, but she can also have this gamete, right? Small m, whereas the male has only the large M and the Y. And so we get the same sort of situation that we saw above with the eyes. We would see that out of the possible combinations, there is a 1 out of 4 chance. There's a 1 out of 4 chance right here. That's supposed to be a small m. I guess the m's are not small m. This would be the one that could produce a male child who has muscular dystrophy. But do you notice something interesting? If it's a female child, she's off the hook. There is no way on this cross. What the heck just happened there? Oh, I see what happened. Sorry. My smartboard just went berserk and jumped on me, and I thought that was the bottom of the cross. This is the capital M here. Sorry about that. For some reason, the smartboard has a mind of its own. So there's what we wanted. And I'm talking about this option down here, right? That's the male that carries the small m recessive allele. So there's a 1 in 4 chance, if you're a boy, for males to have 4 males. But there's a 0% chance for girls. Females in this mating, even if the mother does carry the gene, will not catch it. The only way the female could catch it is if the mother herself had it, and you'd know that. Her phenotype would display the condition. So here's a female who doesn't have it, but might carry it. And so if she is a carrier, then there's a 1 in 4 chance that a boy born of this family would have this disease. In fact, for the boys, if you think about it, if you look at, see again it jumped. If you look at this side of the table, this is the boy side, right? XY, 50-50 chance. If you have a male child, there's a 50-50 chance it will have muscular dystrophy. But if you have a female child, there's a 0 chance it will have it, although there is a small chance, well, there's a 50-50 chance that the girl will carry it on, pass it on. So you see with these sex-linked traits, it's often the male who ends up suffering from the disease far more often, because there's just far more likelihood that the male is going to carry that trait because he doesn't have the extra allele on the Y chromosome. There are all kinds of sex-linked examples. Here are some of the more famous sex-linked examples. Examples of sex-linked traits in humans. Well, we've already seen that muscular dystrophy is one. And it's linked to the X chromosome. It's also possible to have something linked to the Y chromosome. If it's linked to the Y chromosome, it tends to only affect the males because obviously only the males, well, it's kind of the reverse. The males get the option of having the 2 alleles, but the females don't have a Y chromosome. So their homologue is the X chromosome, so they're going to have fewer chances of getting the healthy allele. So muscular dystrophy is X-linked. What are some other? Hemophilia. Hemophilia is a disease where your blood does not clot properly, and that's an X-linked trait. There are more men who suffer from hemophilia than women. Then there's a whole bunch of other ones. Red-green color blindness is a common one. Red-green color blindness. What that means is you can't distinguish the difference between red and green very well. So color deficiency. Color deficiency in your vision. And that's also an X-linked trait. This textbook, in your textbook, I think they talk about hairy ears. This is not really a disease, but hairy ears is Y-linked. It's a trait that's on the Y chromosome. This is having lots of hair growing on the outside of your ears, right? Typically men, we see have this condition, but depends. So the trick becomes, how do you determine whether or not someone is carrying a disease? So remember we went back to this mother here. So how do we know, if you have a mother, whether this is her genotype or her genotype is normal? Because in both cases, she'll look the same. Well, you could do genetic testing. We now have the capability to do testing to figure that out. Or, you could look at the family tree and go back and try to find out what the chances are of that occurring, right? But nowadays, we would probably look at the genetic testing option. And so that leads us to our next sort of thing. Okay, so that's all the linkage that we want to talk about. The next thing I want to talk about is kind of something a little bit different, but it's also sort of related to this. So, maybe what we'll do is we'll stop this video, we'll do a second video.